Hello ladies and gentlemen and fellow biologists, and welcome back to another video. Today this is a very special episode because we are beginning a series on metabolism, which is one of my favorite topics in biology. Basically the sum of all chemical reactions that happen inside the cell, and it's a bridge between biochemistry, organic chemistry and physiology. We are beginning with photosynthesis and not glycolysis like in most textbooks, because I think it's really important to answer one fundamental question, which is where most of energy in ecosystems and living organisms comes from. And I think you cannot start with glycolysis because where does all that glucose comes from? And before I begin, I just like to say sorry for you guys because I messed up my upload schedule. I've been moving places. I moved to Lisbon recently because I'm starting my master's degree in biomedical research, and you need to apply to scholarships because, I mean, I'm not a rich biologist, by the way. So without further ado, let's dive in photosynthesis. So, today we are discussing where most of the energy in living organisms and ecosystems actually comes from. And the answer are primary producers, also called autotrophs, such as algae, higher plants, and some bacteria. And all they acquire their energy from sunlight, and therefore we are going to discuss the light absorption mechanism and the reactions that convert light energy into chemical energy. So, since in this series on metabolism we are going to talk a lot about energy, we need to define it. We are going to just give you a conceptual definition. And if you do not associate metabolism, photosynthesis with the Oppenheimer movie, hopefully you are going to do that at the end of this video. And that's because this movie is all about energy and humanity taming the energy of the atom to build, unfortunately, weapons of mass destruction. But this is a very neat way of introducing the physical definition of energy as the capacity of a physical system to do work. In physics, work occurs when the energy is transferred from one physical system to another via force acting between the two through a distance. But this is a very, very abstract definition, so perhaps you are more familiar with energy defined by a biologist as food. And food is just a shorthand for chemical energy stored in organic molecules, or molecules that contain carbon. And essentially the energy can be used to do work, and the carbon in the backbone of these molecules can be used to assemble body parts. And the question now reduces to how do organisms obtain their food? And there are essentially two strategies, and we are simplifying it here. We have the heterotrophs, or the consumers, which cannot produce their own food, and therefore they obtain it from other living organisms, and the autotrophs, or producers, which can obtain their own food from non-living sources. So essentially, what this amounts to is that when you go to the supermarket and you buy meat or rice, you are essentially buying solar energy converted by autotrophic cells into chemical energy. So I think it's a very neat thought to have. And the question now is, of course, how do plants convert light into food? And to do that, we need to know what light is precisely. And of course, light can be defined as an oscillation in an electric and magnetic field, and the energy on this wave is stored in these teeny tiny packets of energy called photons, also called by physicists quantum of light. And of course, not all waves are the same. Some have higher frequency, as you can see on this extreme, and therefore higher energy, lower frequency, so higher wavelength, so lower energy. And of course, remember that the energy on this wave is stored in a photon, and you can use this formula to calculate energy on these teeny tiny packets of energy. So, but for our intents and purposes, you just need to recall that red light light has a lower energy, and that purple light has more energy. So, and this is important to understand the following experiment because it's very ingenious indeed, and it basically was used to determine the efficiency of photosynthesis in function of multiple wavelengths. So different colors, and basically what the researchers did was put some photosynthetic algae here in line and divided the different wavelengths by using a prism. And you perhaps are familiar that in photosynthesis oxygen is produced, and so if the photosynthetic process is being more efficient, more oxygen is produced as a byproduct, and that is what we see here exactly. They used bacteria known to migrate to regions with higher concentration of oxygen, and I find this fascinating the way they did it. And we can see here two peaks. Curiously enough, we have a lower photosynthetic efficiency on the green region. That's because most of the green light is reflected back, and so nowadays we actually measure this directly, and you can obtain this kind of graphs to calculate what is called an action spectrum. So, how do plants capture light? Essentially, they rely on pigments, and of which there are two main types. You have the antenna pigments with chlorophyll A and B. You are probably familiar with these names, and they are the primary gatherers of energy. And they are slightly different, and therefore result in slightly different absorption spectrum that are sufficiently different to complement each other's range of light absorption. On the other hand, if these chlorophylls are not able to capture all the wavelengths, you can use the carotenoids to absorb wavelengths not absorbed by these antenna pigments to have a broader absorption spectrum. So, where do these pigments exist inside the cell? They exist in these organelles that are very complex called chloroplasts. You have an outer membrane here, you have the inner membrane, and the space enclosed by the inner membrane called the stroma here. So, okay, and then inside the stroma you have these thylakoids, which are again membranaceous bags, so to speak, and they have a space inside which is called the thylakoid lumen, and this is very important for what we are going to explore next, because we are going to explore what relies on the frontier between the stroma, or the outside, and the inside of the thylakoid, called the lumen again. And here is the exact same representation, the stroma and the thylakoid lumen. And here we are going to find our pigments inside photosystems 2 and 1, which are complexes of proteins and pigments, and they integrate electron transport chains, as you can see on this slide. Of course, this is very confusing at first because there are multiple different elements in these chains, but I want you to focus on the main idea of photosynthesis, which is converting light energy to chemical energy, and specifically HTP and NADPH, which are carriers of energy inside the cells. And you can think about them like power banks to charge your phone, but here to furnish energy to multiple cellular processes. So basically what happens is that when you shine light, electrons removed from water by the water splitting complex are energized and pass through multiple constituents on this chain, and ultimately end up in a NADPH. And at the same time, electrons traverse these different elements. Protons are pumped to the thylakoid lumen, so that the concentration of protons on this side is higher than the outside. And since you have a gradient, what happens is that when you open a bridge here between the two spaces, protons are going to flow from the higher concentration side to the lower concentration side, and this flow has enough energy to essentially synthesize HTP from ADP and orthophosphate. This is a very interesting mechanism indeed, and we are going to see this motive in other types of metabolic processes. But now let's focus more on how these photosystems are able to harvest light energy by focusing specifically on the antenna complexes. And here you have an amplification of what we saw earlier, the antenna complex with multiple antenna pigment molecules. And you see that when light is shined on one of these pigment molecules, the energy is transferred between multiple antenna pigments molecules and until it reaches the reaction center with the special pair of chlorophyll A, and then it is finally able to transfer electrons between the different elements in the chain. You should notice that the transfer of energy between different antenna pigment molecules until it reaches the reaction center is called photoresonance energy transfer. And what happens is that you have a donor molecule and an acceptor molecule, right? The donor, as electrons that are excited, they relax a little bit, and their energy gets transferred to the acceptor molecule, and so forth between multiple chains in this chain reaction, so to speak. So now we are ready to talk a little bit more about the different constituents in this redox chain, because what happens is that electrons are being transferred, right? And here we are going to talk about oxidation and reduction, because they happen simultaneously, and redox reactions are reactions in which essentially electrons are transferred. I know chemists are not going to like this definition, but we are trying to give you a clearest explanation of photosynthesis. So what essentially you need to remember is that one molecule loses an electron and it is therefore oxidized, and one molecule gains an electron and therefore it is reduced. So this is what happens in this electron chain. Multiple redox reactions are going to occur. So here you have a diagram. You have here the photosystem 2 and the photosystem 1, and we are going to explore what is called the non-cyclic photophosphorylation. I know the name is very complex, but stay with me. So basically, when you shine light upon the reaction center and antenna complex, what happens is in the reaction center, electrons previously removed from water are going to be energized, and they are going to be transferred to pheophytin, then to the phylloquinones, and from these ones to the cytochrome B6F complex. And the transfers of electrons between the photosystem 2 and the photosystem 1 is through plastocyanin, and these electrons are going to be re-energized once more, and then transferred to iron sulfur proteins, to ferredoxin, and finally these electrons are used to reduce NADP+ to NADPH, as you can see here. This is non-cyclic, but there is another mode of operation of photophosphorylation in which we have a cyclic process. Essentially, what happens is that instead of the electrons in ferredoxin going to NADP+, they are going to the cytochrome B6F complex. And they go back to plastocyanin, then to the center, the reaction center here on the complex, the photosystem 1, and it goes in cycles. This is very important to the production of HTP, because remember, when electrons cycle here, protons are pumped to the thylakoid lumen, and this is important because we are not producing NADPH in this cyclic process. In fact, this is independent from photosystem 2. What happens is that we are just producing HTP because we need, for the second part of photosynthesis, 3 molecules of HTP for each 2 molecules of NADPH. So this is a way of producing more HTP. It's a very neat process indeed. So this is called cyclic photophosphorylation because it's actually a cycle, and the electrons get essentially the ability to move around. And so we reached the end of our lesson. This is an exercise to just associate photosynthesis with the Oppenheimer movie once more. And essentially here is the value of the energy released by the first nuclear explosion. And I want you to compare that energy value with the energy captured by plants, specifically trees on Earth during 1 year. So I hope you find it funny. And so thank you for watching. I know this video was a little bit different because it's the first time we are doing a lecture here on YouTube, and I don't know if this is going to work or not. So share your comments below and don't forget to subscribe, because we are making more of these videos to explain in a few minutes, at least 20 minutes, very complex concepts in a way you can understand, and you can ask any questions related to this matter on the comment section below. And don't forget to share with your friends if you are studying this process, or if you are just curious about biology, make some suggestions as well. So this was Daniel Marks, and I hope to see you very soon. And don't forget, as always, to grab your molecular wrenches.